Hello, everyone, and welcome to day 2 of the workshop in computational and mathematical biology. We're going to be talking about 2 different themes today, ecology and epidemiology, and then morphology. I just wanted to remind you of the website and the Slack channel that we have going on and the GatherTown environment that we are exploring and using to deal with poster sessions and have some discussions after the sessions. So please feel free to enjoy that and walk around. The bio garden joins directly with the poster room. So this is where you can see the posters that are up there. Careful not to get too addicted with the GatherTown environment. So I guess there's not a whole lot for me to say, except that we'll start off with ecology and epidemiology, and we'll be switching over to morphology in the late morning and continuing on through the afternoon. And I'll let Alessandro introduce our first keynote speaker for the day.
All right, thanks a lot, Kathleen. So it's a great pleasure for me to introduce Professor David Dern from the Department of Mathematics and Statistics at McMaster University. I've been at McMaster for many years for postdoc and PhD, and follow with great interest the work of the group in mathematical biology, although it might be the first time which I ask direct questions to Professor Dern. So I want to say that the environment at McMaster is fantastic for all the students that are interested in doing graduate school there. It's a great opportunity, and I would recommend to spend time there because it's a great environment with great people in different groups. So Professor Dern, I would say I have been, so I noticed yesterday on my LinkedIn some great news about him. So I want to congratulate him for being named McMaster Faculty of Science Research Chair. And I think maybe earlier on also he won the research prize for 2021 from the KIMES. I don't want to mess up the acronym, right? So congratulations for that. It's a great pleasure for us to have you in the program of our workshop. And I will stop talking now and let him describe his work on epidemics from Black Death to COVID-19. Please, Professor.
Okay, thank you very much for that kind introduction and for inviting me to speak to all of you about some research that I'm very passionate about.
Sorry, Professor Ern, you're muted.
I'm not
sorry.
muted.
Sorry, I couldn't hear you for a second. It was my problem.
Okay, you can hear me now?
Yes, please.
Okay, so just in case some people didn't hear, I want to thank you again for that kind introduction and for inviting me to speak to all of you. It's a pleasure. So let me just share my screen. Okay, so I hope that you can see my screen now. So I'm going to talk about the research I've done mostly over the last 20 years connected with infectious disease dynamics. And I'll go all the way back to the Black Death, though that's not where I will start. So I'm going to begin by talking about work that we've done in my group on understanding recurrent patterns of epidemics and of things like measles and mumps, rubella, that sort of thing. And then I'll probably spend the largest amount of time talking about that. Then I'll say a bit about work going back to understanding the dynamics of the Black Death. And then I'll end with a brief discussion, since it seems impossible to not mention what we've been doing over the last year with respect to COVID-19. And I'm sure you've all heard many, many talks about COVID-19. But I wanted to say a little bit about what our group in particular has been doing and how that's been connected to policymakers. So let me begin then with understanding patterns of recurrent epidemics. So there are many great examples of such things, and one of the most famous is what I'm showing here, which is the weekly cases reported cases of measles in England and Wales over the second half of the 20th century. And this is a very interesting pattern. It's the number of cases of a given disease in the same place, but the pattern changes it from being initially roughly annual epidemics to then being only big epidemics every second year, and then it becomes quite irregular. And so a classic question is, why is this pattern of recurrence so complicated? Can we understand it? What is the cause of the changes or what are the causes of the changes in frequency over time? So just to make that a little more precise, we can, if we look at each of the temporal segments over which the dynamics seem to be roughly stationary, and look at the frequency contact with a power spectrum, then we can be precise about what it is we're trying to explain here. So initially, the dynamics were visually strongly annual, and there's some power at 2 years. So it's a mixed, strongly annual, but some 2-year power. Then during this period from around 1950 to 68, it becomes overwhelmingly a 2-year cycle with some small annual epidemics. Then it becomes quite irregular, and there's a lot of power at 1 year, and then there seems to be frequency content at about 2.5 years. After this, where the numbers of cases is very low, the vaccination level for measles was so high that there was virtually nothing. Okay, so what is responsible for the changes in frequency contact over time? I mentioned vaccination at the end, and that may have something to do with it, but it's not at all obvious why increasing vaccination level would change the pattern of recurrence. That would explain, for example, the overall reduction in the number of cases in this period, because vaccination started in 1968 in a big way in England. But it doesn't explain the change in periodicities. So what's going on? Well, the way that mathematical epidemiologists have approached this is to try and build a simple mechanistic model, which many of you will have seen before, but I'll explain here. The idea being to capture in a simple way the process of disease transmission and what we would expect to see at the population level. And I'll comment too that one could look at a time series like the one that I showed you and then use statistical time series methods to try and project forward, but that tends to be quite poor at predicting changes in patterns of epidemics. It's much better at predicting the behavior a short time in the future. So this is called the SEIR model because individuals are divided into several compartments. They're by disease status, they're either susceptible to the disease, exposed, meaning that they are infected but aren't yet infectious. So they have a latent period. Then they're infectious, or after recovering from the infection, they're immune. So the recovered class contains individuals who are immune. And I'm thinking about measles or similar infections for which the mortality rate is extremely low. And so I'm ignoring mortality from the disease, but not from natural causes. So there are births that occur, and so new susceptible individuals enter the system from being born, and anybody in any disease class can die of natural causes other than disease. And because we're thinking about infections for which there is a vaccine and has been for a long time, we model vaccination as catching individuals before they enter the susceptible pool and taking them directly to the recovered class. Okay, so then the idea is that we imagine that the population is homogeneously mixed, and so transmission occurs because susceptible individuals meet infected individuals, and then with some probability, the susceptible becomes infected. And so we have new susceptibles entering the dynamics, infections occurring, people ending their latent period, and then this is the number of infectious individuals who eventually recover. So the parameters here are the birth rate, the death rate, the proportion of the population vaccinated if there's vaccination going on, and then the transmission rate, beta, and then there's sigma and gamma, which are rates, but they're easiest to understand in terms of their inverses. So the mean latent period, the mean time between when you get infected and when you can infect somebody else is 1/sigma, and the mean time that you're infectious, ignoring the possibility that you die during an infectious period, is 1/gamma. Okay, so here is a simple model that in principle can explain what's going on. If we analyze this model with standard tools of qualitative theory of ordinary differential equations, what do we find out? What are the possible dynamics? Well, very easily, we determined that there are exactly two possible equilibria. Equilibrium at which no disease is present, which we call the disease-free equilibrium. So I and E are both 0 at that equilibrium. Alternatively, there is potentially an endemic equilibrium, that is an equilibrium at which there is always prevalence of disease. Now, an equilibrium isn't the type of solution of the model that we might hope for, because I just showed you a time series that showed periodic behavior. So are there periodic solutions to this system of equations? Or are there chaotic solutions which could explain the irregularity that I mentioned? The answer is no. There are no such solutions. And the easiest way to show that is or at least a helpful way to show that is to introduce a parameter that will be familiar to you, I'm certain, from reading the news over the last year, and that is the basic reproduction number, which I'll explain in a moment, can be written in terms of the parameters of the model that we wrote down. And R0 is the expected number of infections that will be caused by one infected individual who enters a wholly susceptible population. So it's if you enter a situation where everybody's susceptible and you're infected, how many people would you expect to infect? And that's it's a dimensionless quantity that's easy to understand and is a very convenient way to classify solutions, formal solutions of the model as well. So how do we find R0? Well, biologically, we can just think in terms of the meaning that I just described and related to the parameters. So if we assume for simplicity, which I will, that for the moment that the birth and death rates are the same and that there's no vaccination, then R0 is going to be the rate at which transmission occurs times the probability that you actually survive the latent period. So sigma was the rate at which you leave the latent class. And so there's a small probability that you die during that. But if you survive, then you will be infectious for a period 1/gamma plus mu, or this is really the amount of expected time in the infectious class, taking account of the fact that you might die of natural causes while infected. Okay, so that's a formula for R0, which, because mu, the death rate, is very small, is approximately beta over gamma, or beta gamma inverse. Okay, so because the mean lifetime 1/mu is much greater than the mean latent period and the mean infectious period, it's approximately beta gamma inverse. So we can also derive this quantity mathematically by asking what the boundary is between stability of this endemic equilibrium. And if we do that, we find that R0 equals 1 is the stability boundary if we define R0 by this formula. So as we can find the formula for the stability boundary, it's this being greater than or less than 1. Okay, and it turns out that this formula, as a matter of interest, beta gamma inverse applies much more generally to more sophisticated models if we interpret beta and gamma appropriately as matrices that arise from a more complicated model. Okay, the outcome of the analysis is that the endemic equilibrium is globally asymptotically stable. Regardless of initial conditions, you will end up at that equilibrium, provided R0 is bigger than 1. So provided the average, on average, you infect more than one person, you will end up there. Otherwise, so if you on average infect less than one person, so the number of cases must be going down, you will converge onto the disease-free equilibrium. Okay, so now I said we were looking for periodic solutions, and it does turn out that the endemic equilibrium is approached by damped oscillations. And so you might say, oh, okay, well, maybe we're just seeing damped oscillations. But that's a weak potential explanation, because the observed epidemics that I showed you are undamped. They do not damp out. Okay, so we started with a simple model that doesn't on its own explain the patterns that I showed you what does. Okay, so we must be missing something in our model. What is that? So one thing is that by writing down differential equations, we've essentially assumed that the population is infinite, or that it can be infinitely divided. Whereas, of course, individuals come in units of integers. So really, we should be taking into account that finiteness and the fact that the process isn't purely deterministic, that there are random processes involved in contact and disease spread. So it turns out that if we recast the model as a stochastic process, then it can be proved that in the limit that the population size goes to infinity, we end up with the differential equations. But the population size isn't infinity, so the question is, what is the behavior for realistic population sizes? And we can explore that in an easy way with simulation. It's much harder to do it analytically, but with simulations we can easily look at what happens for population sizes and parameters that are relevant to the real world. And there's a standard simple algorithm for doing that, which I won't go through in detail, but I'll show you what the outcome is. So if we take this SEIR model parameterized for measles and solve the differential equations, we get this red curve. So the red curve shows the damped oscillations onto the endemic equilibrium. Measles has a very large R0 of 17, and these are the mean latent and infectious periods. And this is for a population of 5 million. Even with a population of 5 million, when we look at the black circles show a realization of the stochastic process, even with 5 million individuals, the oscillations do not damp out. Okay, well, that's a really important result that has been known since the 1950s due to Morris Bartlett. And so the transient behavior that eventually damps out in the deterministic model and converges onto an equilibrium never damps out stochastically even for realistic population sizes. So that's good. That means we actually have a simple model that creates recurrent epidemics. However, the period between those epidemics is always predicted to be the same. And yet what we saw were changes in the length, the time between epidemics. We saw annual, biennial, and more irregular epidemics. So this isn't quite enough, but it does seem like potentially a step in the right direction. Okay, so we can't explain the observed time series that I showed you of measles, but at least we can get damped oscillations. That's something. Undamped oscillations, excuse me. Okay, so we must still be missing something. What's that? Well, I assumed implicitly, actually, that the transmission rate, and in particular the contact rate among individuals, was constant, and that's false. Okay, in England and Wales, where I gave you the example, their kids who are the individuals who are most likely to get measles are in schools, and schools are not in session constantly. And so the seasonal change in the transmission rate when kids are aggregated in schools or not can be very important. So a simple way to model that is to replace the constant transmission rate, so that's the beta here in the equations, with a sinusoidal oscillation. Okay, so that's a simple way to incorporate, at least conceptually, the idea of seasonal change in the contact rate due to aggregation in schools. So what happens if we do that? Does it make a big difference? Well, those of you who've studied nonlinear dynamics will know that it certainly does, because we have a nonlinear system. There's that beta SI term, which makes the system nonlinear. And when you force a nonlinear system, the natural time scales can resonate with the period of, with the forcing period. And that can lead to very exotic behavior. We get a very rich dynamical system, similar to the way that tapping a damped pendulum, with a constant frequency, hammering it, will cause very complicated behavior. Okay, so what types of behavior exactly do we see? This bifurcation diagram summarizes what we can get out of the sinusoidally forced SEIR model, parameterized for measles, as a function. On the x-axis here, we have the amplitude of the seasonal oscillations, the amplitude of the seasonal forcing. So the amplitude of that sinusoid. So if the amplitude is very close to zero, we get an annual cycle here, which means 1 point, because what we're doing is strobing the system once a year. And we always come back to the same point at that same, this is the proportional prevalence at the same time of the year. For somewhat higher forcing, we get a biennial cycle. That is, in alternate years, we come back to the same point. And for some ranges of seasonal forcing, we get a wide band, which indicates chaos. We never come back to exactly the same prevalence at the same time of year. So that gives very irregular behavior. Okay, well, this is actually suggestive that we might have something that could explain the dynamics. Okay, so does it explain the picture that I showed you? Well, this model with sinusoidal forcing does produce recurrent, undamped epidemics of all the frequencies that we've ever seen in any measles time series, not just in England and Wales, but anywhere. It produces chaos, and that can explain the irregular behavior that we saw after 1968 in England and Wales. However, I'll note that if that's the correct explanation, then the time at which those irregular dynamics began would not be predictable. Okay, on the other hand, this model also predicts that the virus would have gone extinct almost immediately. And why is that? Well, if we look again, sorry, I went back further than I intended. Whoops. There we go. If we look at even here at the chaotic regime for modest seasonal amplitude, this is a log scale, and the proportion of the population infective is going down to 10 to the minus 20. So that's nobody. And so if we are in that regime, then in fact, we would have predicted for any reasonable population size that the system, that the pathogen would have gone extinct. So that's not good. So we must still be missing something. What might that be? Well, we said that contact rate was higher during school terms, but I just quickly said, well, let's just model that as a sinusoid. But it's not a sinusoid. I mean, suppose that instead of just for convenience modeling seasonal oscillation as a sine wave, we actually correctly model it as high during school terms and low otherwise. but we can at least numerically, change that in the model and see what happens. And if we do, rather than getting all this crazy zoo of dynamics, it turns out that we always predict a biennial cycle for measles epidemics everywhere. Well, that's not what we were hoping for. That was sort of made things too simple in a sense. So we've got undamped biennial cycles now. Where does that leave us? Well, it turns out that if you look at the agreement between this model and that period of biennial oscillations that I showed you for England and Wales and in various other places, it's really good agreement. And so that seems like too much of a coincidence for this to be wrong. So what's going on? We must be missing something else. What is that? And the key insight that allows us to understand this time series and many others is that the effective reproduction number, the effective R0, is actually changing over time. So if you imagine that you estimated R0 at a time when the birth rate in the population was new, say, it turns out that if the birth rate changes, say to new prime, then the dynamical effect, that is the pattern of recurrent epidemics that you see, is equivalent to if you had changed R0 to R0 times new prime over new. And that's easy to prove just from a scaling transformation and then looking at the equations. Okay, but this is an important insight because it generalizes to any change in the rate at which susceptibles are recruited into the population. So for example, if the birth rate changes to new prime and the vaccination level proportion vaccinated changes to P prime, then the dynamical effect is equivalent to changing R0 to R0 times this factor. And because we have data on birth rates and vaccination levels, that means that we have a way of predicting changes in the dynamics. But with using a bifurcation diagram that varies as a function of R0 rather than the seasonal amplitude. Okay, so changes in the birth rate, the vaccination proportion, or in the contact rate, the transmission rate directly, all map onto this same axis. And so we can summarize the possible behaviors with this one parameter bifurcation diagram with respect to R0, or equivalently the transmission rate. And then we can predict the transitions that occur by mapping them onto this diagram. So here is such a diagram. The horizontal axis is the mean transmission rate, or equivalently R0, which is proportional to that. At the time when, in the 1960s, when R0 was measured as about 17, that corresponds to a mean transmission rate about here. And what this predicts is that there should have been a biennial cycle. As I mentioned, there's great agreement. But then an increase in birth rate would push you up here, potentially seeing an annual cycle, or or a reduction in birth rate or vaccination would push you down here, where there are many coexisting stable cycles. And what's shown above here is that these are basins of attraction. So the initial proportion susceptible versus initial proportion infective. And as you as you reduce the effective R0, the basins of these basins of the different stable cycles become more and more intermixed. And so with a little bit of noise, you tend to jump around, and that could explain the irregular behavior that we saw. Okay, so how well does this work out? Well, we can apply it, for example, to a bunch of data that we have for measles in various cities. So here, for example, is measles in London, England, the second half of the 20th century. And what's shown in red is the birth rate in London, the solid red line. And above, the colored lines correspond to the regions of this bifurcation diagram where these various cycles are stable. And so in London over this period, we predict the biennial cycle, and that's indeed what we see. Earlier on, we're actually in this region where there's both a biennial cycle and a and an annual cycle, and I've marked it as annual because that's what actually occurred. It could have been either one, but it's an annual cycle that occurred. Later on, we moved into the region where there are many stable coexisting cycles, and that's where we saw irregular behavior. Similarly, in Liverpool, another British city, but the dotted line here is actually Liverpool's birth rate, which was higher than London. And throughout this earlier period, Liverpool was in the region where there was both an annual and biennial cycle, and we happened to see exactly an annual cycle that whole time. In New York City, and a sort of opposite type of population level experiment was done, because this has been the pre-vaccine era, but during the Depression, the birth rate was much lower, and we see irregular behavior early on, and then when the birth rate rose, we see strictly biennial cycles, again, agreeing with this model. So this is really encouraging, and the similar story in Baltimore. Okay, well, that's great success for measles. With that in hand, we might say, well, what about other notifiable infectious diseases for which we have data? Clearly, we can play the same game. That is, we can apply exactly the same model, but with different parameter values, different mean latent periods, different mean infectious periods, and so on. What happens when we apply exactly this analysis? Does it explain recurrent epidemics for rubella, for chickenpox, whooping cough, which are other infections that were extremely prevalent in the 20th century? Unfortunately, no. It doesn't work. So what I've just shown you fails when we apply it to all of these other diseases, and that's extremely disappointing. So things seem to be going very well, but for these other diseases, when we parameterize the SEIR model, it always predicts an annual cycle. There aren't all these different possible behaviors, even with seasonal forcing. So what is going on? Just to show you how bad this is, here's an example of rubella. I'm standing in Ontario at the moment, actually. That's where I'm based. This is weekly cases of measles in Ontario during a lot of the 20th century. And the dynamics are much more complicated than a simple annual cycle. There are periods of annual behavior, it seems, but then very large epidemics once in a while. And then later, coincidentally, when the birth rate rose, the dynamics change. And on the right-hand side here, I have marked the power spectrum in this early region in black and later in red. And we can see that early on, there was power at a much longer time scale. And then later, it's really only at the annual cycle. And there's similar changes in whooping cough in the UK, which again, the model predicts, according to the analysis that I just said, just an annual cycle. And instead, what we see in these different periods is a single main period, which is not annual, around 2 years or around 3.5 years. This is before and after vaccination kicked in whooping cough in London. Okay, so these time series of notifiable diseases are showing behavior that is not predicted by the attractors of that model. Okay, so what do? As mathematicians, we sort of scratch our heads and try and figure out what could be missing in our model, or what is it that we're missing? Are we missing something in our model, or are we missing something in the analysis? So remember that the first step in changing the SEIR model was to realize, oh, wait a second, it's not a deterministic process. We need to think about individuals. We need to think about demographic stochasticity. And in fact, that's really important here. And I brushed that under the rug when I was talking about seasonal forcing, because I didn't need the demographic stochasticity to generate recurrent epidemics anymore. But that turns out to be extremely important, because stochasticity sustains the transient behavior. And it turns out that if you apply linear perturbation theory to the attractors of the SEIR, the seasonally forced SEIR model, so this is looking not just at damped oscillations around an equilibrium, but also damped oscillations around more complex attractors, that actually allows us to explain what happened in these other diseases. Okay, because it turns out what we're seeing is exactly the expected behavior from the transients on the approach to these attractors. Okay, so given that that's true, of course, then we get even more ambitious. So we can try to explain not just the main spectral peak, which is what I was emphasizing when talking about measles, but all the spectral peaks in the data. Maybe we can do even better. We can predict the resonant peaks, perhaps. That is, by resonant peaks, I mean, in the power spectrum, I mean the ones associated with resonance with the period of forcing. That should come out of the asymptotic analysis, the bifurcation diagram. And then the non-resonant peaks, which arise from the transients, non-resonant may not be the best word, but that's what we've been using. Maybe we can predict that from this linear perturbation analysis, and that turns out to work. And here is an example from quite a few years ago, looking at the predicted non-resonant power spectral peak for a bunch of diseases in different places versus the observed, and there's very strong agreement here. So we seem to be getting it right. And I'll mention too that for the resonant peaks, we get those exactly correct always. That's less surprising because they're constrained to be an integer. Okay, so that's great. It seems we can do well at predicting both spectral peaks. So let's get more ambitious. Maybe we can predict even more detail. Just like a physicist who keeps trying to get to 11 decimal places. Sadly, we won't get quite there. So can we predict the magnitudes of the spectral peaks, not just what periods they occur at, but how large they are? And to see that there's an issue here, I am showing you examples of weekly case reports of chickenpox in Ontario versus Manitoba, 2 Canadian provinces. And at a glance, these time series look extremely similar. They both have strong spectral peaks at 1 year, but there's another spectral peak, and the relative magnitude there is very different for these two diseases. And the reason would appear to be that there is a great deal more demographic stochasticity in Manitoba, where the population size is about 1/10. And so we would expect there to be more evidence of transient there than in Ontario. And that does seem to be what's going on. And so just summarizing everything that I've said about these recurrent epidemics of childhood diseases, we seem to understand their patterns very well using a simple mechanistic model and some, well, non-trivial analysis of the seasonally forced nonlinear system. We predict the resonant spectral peaks perfectly, the non-resonant ones very well, and we find that the population size is the key determinant of the relative magnitudes of those peaks. And that's confirmed with stochastic simulations. And there is some analytical work in that direction as well, which is perhaps for another time. Okay, so what do we do with this good theory? Well, what we've been doing in the last few years is trying to apply it to even longer time series and see if we can continue to explain them as effectively. So here, I'll just very briefly note and not go through any details. A PhD student of mine went to New York City and extended the time series by digging through archives back to 1891. The other New York time series that I showed you was just since 1928. And cutting a long story short, the theory continues to work. Instead of showing a simple power spectrum, we're showing the wavelet spectrum, which at each time point shows the spectral peaks in the time series. There's extremely good agreement between the predicted resonant peak, which is marked with circles here, and the predicted non-resonant peak, which is these white bars with the time series. And that is good news. I should say, except in the period over here, there's no these are very long predicted periods during the biennial cycles, and those are not detected. There's too much noise to detect very long period transients, which is why the measles theory worked so well without doing the perturbation analysis of the attractor. Okay, in order to do this work with these longer time series, there was more that we had to do. We had to worry about the fact that, in fact, the pattern of seasonal forcing is not the same. And a couple of recent papers with students established how to expand the theory to take account for that, which we did. Actually, and I didn't explain, but this colored plot in the middle is over time an indication of the volumes of the basins of attractions of the different attractors. And so, for example, here, red is the biennial cycle, and that's very strongly what we see, whereas in other regions, there are multiple attractors, and we might have been approaching any one of them. Okay, and I'll just mention also that we're in the process of applying this theory to the longest time series anybody has ever studied, which is that for smallpox going back to the middle of the 17th century. This picture is from a paper just published with a student, Olga Krylova, who worked very hard to understand and describe these patterns and how they're related to changes in historical policies on control and the introduction of vaccination for smallpox and so on. And we're in the process of applying the theory to this. Okay, so that's the main section of my talk, and I want to briefly talk about two other areas. So the next is, you can probably tell that I'm very passionate about looking at old data and trying to make sense of it. And the earliest old data that I have been able to get my hands on comes from the city of London, England, and that's the bills of mortality. And so here is an example and showing on the left the list of causes of death and the number of people who were thought to have died from these various causes in this given week. And if we look at a few of them, so somebody died of being frighted, 3 people of grief, lots of other interesting causes, but over 5,000 people in a single week in a city of only about 500,000 at the time died of plague. Okay, so plague is a very interesting thing to study in these old records. But the first plague that we have any hope of understanding is the Black Death from 1348. And the types of records I just showed you don't go back that far. So we do have back to 1563 those types of records. A little further back to 1538, we have parish register records. But deaths started being registered in England only in 1538. Before that, we have no chance of getting mortality records, but we can look earlier, by looking at last wills and testaments and counting up the number of wills written on given days, which we have done. And what happens when we pull that together is we look in the 14th. I couldn't hear what you said. My watch thinks it's talking to it. When we look at the 14th century, this is the pattern of wills, and I've highlighted in yellow the years when historical records indicate there was a plague epidemic and there were more wills written. This is a log scale, so these are quite large jumps. In the years when we know there were plague epidemics, so people were probably writing their wills because they were scared or knew they were about to die. Later, we also have wills, of course, which we've pulled together. And then we have the other sources of data from the parish registers and from the bills of mortality, which specifically say deaths from plague. Okay, and we can look at, for example, in the 14th century, this is the weekly pattern of wills written in 1348, 1361. We can see that this does suggest that we're picking up signal of the actual epidemics. Later on, I'm comparing all 3 data sets in 1625, 1665. 1665 is the Great Plague of London. And all 3 sources are consistent with the wills being a lot noisier. Okay, we'd like to compare the early 14th century with the later 17th century epidemics. But a key question is whether we can really do that when we have only wills in the earlier century. Fortunately, because we have wills later on as well, we can investigate that question. So here is just the Great Plague of London in 1665. This is from the bills of mortality, the actual number of recorded plague deaths each week, which is the best data we have. And here is the pattern of wills written in 1665. And you can see that the pattern is very similar. And indeed, the rate of growth during this phase of growth is very, very similar, suggesting that we could compare the rates of growth of plague epidemics in the 17th century with those in the 14th century and see what we learn. So we'd like to do that. And I'll emphasize that this is an important thing to do. We want to just compare the epidemic curves because there's a lot about plague, which I haven't told you and which we don't know. So at least we can't verify for these old data. We don't want to assume anything about the mode of transmission for plague when we look at these data, because we don't know if we're looking at epidemics of pneumonic plague, which is caused from human to human transmission of the pathogen Yersinia pestis, or whether it's bubonic plague, which arises from rats being infected and there being spillover to humans. And the way that you model those are different. We can, though, just from the data, compare the rates of growth. So let's see what we can learn from that. Did 1 century give rise to faster epidemics than the other? So the naive way to estimate a growth rate is just say, well, okay, let's look at the epidemic on a log scale. This is the Great Plague again on a log scale. And we'll just look at the period of what appears to be exponential growth and fit a straight line to it. And the slope of that straight line is the rate of exponential growth. But I'd identified this as the period of exponential growth. Early on, there's obviously some noise. And you might have identified this as the period of exponential growth, which would also be reasonable. And we would each get quite different estimates of the rate of growth. So that's a bit of a hairy problem. We actually need to have a statistically rigorous way of estimating the growth rate and a confidence interval so we can make comparisons. And cutting a very long story short, I'll say that the solution to that problem is, instead of just fitting an exponential curve, we fit a saturating curve, which has exactly the same initial exponential behavior. And it turns out by doing this, we can reliably estimate that early exponential growth, even from very noisy data. And we've tested this very extensively. Again, that's another talk to go into the details of that. The upshot is, which I think is very interesting, is that the later plagues in the 17th century had a growth rate that was about 4 times faster than in the 14th century. Okay, that's a big difference that was not expected. And rates aren't as easy to understand as times. And the equivalent time that it takes for the number of observed wills or cases to double was around 45 days in 1348, but only around 11 days in 1665. It's a huge difference, much faster epidemics later on. So that's an interesting discovery. And it obviously, then we can ask, well, why did this happen? Was it faster later on? And there are all sorts of possible reasons. The pathogen could have evolved to be more infectious or have a longer infectious period. There were changes in population density and social structure. The weather was different. What's known as the Little Ice Age occurred during the later part of this period. And we don't know if we're looking in one case at epidemics of bubonic plague and the other at pneumonic plague. On that last question, I can say just a bit more, which I think is quite interesting. And that's that if we're willing to make the assumption that the plague epidemics in the 14th century, if they were pneumonic, were identical to modern pneumonic plague. Pneumonic plague still occurs around the world ever so often. And so we know a lot about it. If we use the parameters we estimate from modern data, then we have enough information to actually estimate R0 for the 14th century and consequently the expected final size or attack rate of the whole epidemic. And if we do that, then for the 14th century in London, if it were strictly a pneumonic plague epidemic, we would estimate that about 20% of the population in total was infected. But we know from historical records that something like more like 30 to 50% of the population of London died in 1348. So that's really inconsistent, suggesting strongly that the early plagues were not just primarily pneumonic plagues. And that's not the world's strongest inference. I think it's interesting, but remember that we're managing to make this inference about what the mode of transmission of an epidemic was in the 14th century. And we're doing that by counting the number of wills written by individuals who had some property to leave. And so I think that is quite a feat and is interesting and certainly leads to more questions that I hope we'll do better at answering in the coming years. Okay, so finally and very briefly, I want to say something about the epidemiological modeling that we have been doing during the COVID-19 pandemic. And it won't surprise you that people like me were contacted very early on by governments to try and provide forecasts of what might be happening and how to help with planning and policy. So just to take a look, so SARS actually entered our population in 2003. The first SARS coronavirus gave this worldwide epidemic, but it was a pretty small, really. Those of us who remember it well found it very alarming, but there were in total only about 8,000 cases and about 800 deaths. Had that spread into a worldwide pandemic, it would have been much worse than the current one because the mortality was about 10%. Today, this is up to last night, the number of reported cases worldwide of SARS coronavirus 2, close to 175 million cases and 3.7 million for 3.8 million cases reported, and probably at least 3 times that actual numbers of deaths from COVID. So a catastrophic experience over the last year. Here is the same data on a log scale showing the SARS coronavirus 2 epidemic, and this on the same scale, just lined up, is the equivalent SARS 1 from 18 years ago, which was minuscule in comparison. And I've shown growth rates and doubling times. So initially, when it was just in Wuhan, primarily, or actually this is beyond Wuhan, when it got a bit further out, we saw very short doubling times, around 5 days. And then because of when it came into other countries, it wasn't quite as fast if they engaged in control. Okay, so I'm in Ontario. We've been doing a lot of work, in particular, trying to help the Ontario government understand what's going on and what policies might be helpful. So here is the pattern of the epidemic in Ontario with the growth rates, with the fits of initial growth and doubling times on top. So initially, it was spreading very fast with less than 4-day doubling time way back in March of 2021. At the moment, we recently had a large third wave induced by a variant, but it's looking good for the moment if you just look at this case series. So how do we make projections that are useful to governments? Well, fortunately, we have much richer data than the kinds of things that I showed you, even for measles, certainly for the Black Death. You know, we have daily counts of all sorts of information about these epidemics. We even know the daily numbers of vaccine doses administered since there's been a vaccine. And we have all sorts of detailed measures about things like mobility patterns in addition to weather and so on. And we know exactly when policy changes were made. So with all that information, that we can do well with forecasts, but we have a much harder problem because we actually are trying to forecast, not just trying to understand data that we've collected from the past. Okay, Well, we do that by expanding that SEIR model to include more compartments for the types of things that data that we observe. And then we simultaneously try and fit all that stuff together and then make a prediction for the future. And we have to interpret any such prediction we make with a lot of caution, because even with all these data, it's still an oversimplified model, and we cannot take account of all uncertainties. And even if we could, we'd have confidence limits because of stochasticity. Okay, so what happens with those caveats? What kind of projections can we make? Well, way back in mid-February, we made the following projection. So the black dots are the observed data in Ontario, and we fit a couple of scenarios going forward in time from the date of the forecast, which is here in late February. If we just naively fit our model, we would project the blue curve if the province chose to open up, that is, stop I forget what they called it at the time, but the reducing contact measures that were in place. There would be a little rise, but it would be modest eventually. But if we took into account the information we had about the spreading alpha variant at the time, wasn't called alpha variant then, we projected that if they opened, there would be this huge rise. And unfortunately, the fact that the cases were going down were more persuasive to the decision makers than the model. And so what actually happened was they did open up even earlier than they projected. And then, of course, it did rise pretty much as we projected. Later, as more data accumulated, we could make more precise predictions. And again, they did very well. This is our current forecast. So this is submitted just this week. And so the date of the forecast, which was June the 5th, is here. Again, the black dots are the observed data to which we fit the model. And these circles are the data that occurred since we made this forecast. So it's continuing to follow these curves. And there are several scenarios shown here based on the planned today. The opening is today, in fact, in Ontario. But we don't know is whether that will lead to an increase in transmission rate by a certain amount. So we considered scenarios of 50%, 75%, or 100% increase in the transmission rate. And we also considered possibilities of increasing the rate at which vaccine is distributed. And the key thing here that's going on beyond vaccination and increased contact is that the Delta variant is spreading. And that's what's giving the possibility of a fourth wave here. And the way to avoid that is to increase the vaccination rate. And I hope very much that that's going to be possible in the coming weeks, and that we can avoid the worst of these scenarios going forward. So I've covered a lot of ground and run through some things very quickly, but I hope that has convinced you that if you didn't already know that there's a lot of interesting science and practical public health work that can be done using mathematical models of infectious disease. And I'd like to thank these various collaborators, students and colleagues, and funding agencies as well, all of whom have been important for this work. And finally, thank you again for inviting me to speak to you. And I look forward to any questions. Thanks.
So let's thank Professor Ron for a wonderful talk. And let's open the discussion to Q&A. Are there any questions?
I have one question.
Yes, please.
Regarding the inclusion of birth rates into your model for, I think it was measles. I just thought, if it seems that schools seem to be a big driving factor for the spread of measles. So I thought, would it be more accurate to use, instead of birth rates, the number of children who are starting school for the first time each year? So
that's a really good question. And in fact, that's a subtlety that I chose to ignore, but we actually worry about that. So really what we're doing is, and so we shift the actual birth rate to the date at which kids typically enter school, and we ignore the relatively small amount of transmission that occurs before kids enter, well, kindergarten or school. So that's a really good question. And in fact, it turns out that that means that instead of being a continuous influx of new susceptibles, it happens quite suddenly at the beginning of the school year. And so we've worried about that effect as well. But qualitatively, there's no difference from what I was presenting to you. But thank you, a really perceptive question.
Thank you.
Other questions? Yes, please.
Hi. So you showed that for measles, they are those spikes that they are correlated to birth rate. So I was wondering, because that would suggest that R coefficient for measles is extremely high. Could you refer this as a comparison to current pandemic?
Oh, yeah. So the R naught, I think you mean. So the average number of secondary cases from a given primary case when everybody's susceptible is about 17 for measles. It's huge. It's one of the most transmissible infections we know of. Whereas for SARS coronavirus 2, it's of order 3 or 4. I mean, it keeps getting larger, unfortunately, with these new variants, but it's much smaller. And a relevant, something that that is in many people's minds is what the threshold for herd immunity is. So that means what proportion of the population would you have to vaccinate or otherwise protect in order for the epidemic to end, because not enough people are being infected to sustain it. And in the simple SEIR model, that can be that proportion, that threshold can be written as 1 - 1 over R naught. And so for measles, that means that you've got to reach about 95% of the population, which we do in most parts of the world, but not everywhere. And you have to actually successfully immunize. So that might mean actually, that means double dosing with measles. Anyway, it's a very large proportion. If it's, if let's say R naught is 4, then instead you're talking about 75% of the population has to be immunized. And that's where people come up with this 70% figure that keeps going around. But that's a very crude estimate for a lot of reasons. Not just because the SEIR model is oversimplified, but because it doesn't take into account that the vaccines aren't perfect, and there's a lot of heterogeneity in the distribution of the vaccine and a lot of other issues. And so I think herd immunity, true herd immunity that would cause us to completely eliminate COVID is not on the horizon, unfortunately. So that's a long that went beyond the question you asked, but hopefully of interest.
Thank you. Thank you very much. It was extremely interesting.
I have a very quick question about the result of measles. Like, it's basically kind of a dream type of result, because with the minimal amount of parameter, you explain all the phenomenon. And I was wondering if something like that is hopeful also for what concerns COVID-19, because having that understanding would be, I think, kind of fundamental. I don't know if there is any hope, though.
Well, I think that, no, I think it is. I think a simple model like that would potentially be very successful if you ask the same types of questions about COVID. If you ask, suppose we're past the initial pandemic and we want to understand the patterns of recurrence that we would expect under various vaccination strategies, then we might do quite well. If we actually want to do the type of detailed forecasting that I was talking about, like we really want to know exactly when takeoff is and exactly what the short-term responses are to various policy changes, we need to be a bit more careful than that. Though, I mean, really, the model that we're using isn't that much more sophisticated than the SEIR model.
Thank you. Well, I will stop stealing time from Professor Ron and the next speaker as well. So I want to thank Professor Ron for a wonderful talk, and also to the people which asked questions, very interesting questions. So thanks a lot for your presence here, Professor Ron.
Thank you all very much.